going through the central dogma now, which is replication, transcription, translation, making sure that you have all the appropriate vocabulary and not mixing it up. This diagram of replication happening is pretty neat. You can see all the things we talked about. Semi-discontinuous. You can see most of the enzymes we talked about. Semi-conservative, bidirectional, asymmetric, all that stuff is kind of apparent as this kind of zooms around here. We even talked about the exit exam question where you're asked to use 20 key words in a short paragraph to describe this whole darn thing. It's worth practicing that out. This is absolutely something that students of molecular biology are supposed to know cold, like the back of their hand. DNA polymerases, it's important because today we're going to contrast this with RNA polymerases. Like primase, primase is an RNA polymerase. DNA polymerases have a primer requirement, the 5' to 3' polymerase activity plus an exonuclease activity so they can go backwards. If they're not getting the primer requirement met, they can go backwards and erase what they did and then do the forward activity and maybe get the answer right this time. And so DNA synthesis is said to be high fidelity. The copies of the templates that we're making are almost perfect, except for rare mutations, and we don't want to snuff out mutations. We need mutations in order to have variation in the population. We need the variation in the population so we can survive changing conditions. So these are the general characteristics that all DNA polymerases have. The different DNA polymerases may specialize to one degree or another, or in terms of the timing in the cell cycle, but in general, that's what's true of DNA polymerases. And again, we'll contrast that with RNA polymerases today. Before we moved on, last time we stalled a little bit with replication to see how it can be exploited in the laboratory. So this depiction of the polymerase chain reaction, starting with just one template double-stranded DNA, denature it, anneal your forward and reverse primers, extend those paired primers, and then when you denature that, you end up with now 4 templates instead of the original 2. Anneal your primers, extend, denature, now you've got 8 templates instead of 4. And you start to pile up according to a geometric progression. In just 30 cycles, you end up getting something that is along the lines of 1 billion copies, not of the whole template that you started with, but of the specific region on the template which is located between the primers themselves. You choose the primers based upon what it is you want to amplify. Let's see, on a gel, the results of a polymerase chain reaction can reveal 2 different alleles of a particular site in the genome. That's one way that the polymerase chain reaction can be exploited. In this case, we're not looking at genes, we're looking at the boring spaces between genes because we're looking at places where there are variable numbers of tandem repeats, dinucleotide repeats. Sometimes these repeats are sections of 5 or 10 or even 100. These variable numbers of tandem repeats don't encode information. DNA polymerase does a lousy job at copying them accurately, and so what they provide us with is a lot of variation, even among close relatives. Even twins will often differ at these variable number of tandem repeat sites. And so the father's chromosome, the mother's chromosome may have at this particular site different numbers of those tandem repeats, and on our electrophoresis gel, we can visualize daddy's allele, mommy's allele for that particular site in the genome. And there's tens of thousands of these sites that are known and characterized, and their positions on all the chromosomes is firmly established. And so there's no limit to the amount of genetics that can be done, even with a complex genome. Here's another application of that polymerase chain reaction. And this is exactly like what we are in the middle of doing in the laboratory that we'll finish up next week. Here there is simply a single base difference between the wild type and the mutant sequence in this particular location in the middle of this particular gene. Where there should be a G, instead the G has mutated to be a C. Where there was a HaHa1 site, there is now a He3 site, but not the HaHa1 site. So very frequently, changing any base is likely to alter some little endonuclease target site like this. Now, instead of doing a southern blot, we use PCR to amplify that small region. As we did here, the overall amplomer was just 286 base pairs. All mice gave us that same amplomer because they all have this gene in general. Expose that amplomer to the He3 enzyme, and no normal mice have that sequence, but the mutant mice, their amplomer is cut into 2 smaller pieces. And you can even judge the sizes of the pieces. We measure them very accurately. These 2 numbers add up to that number. We could do the opposite experiment, take the amplomer, everyone has the same size, cut it with HaHa1. Now normal mice cut, showing that normal mice have the HaHa1 sequence, but the mutant mice have lost it. This is also, I guess, a reminder, when we set up our gel next Tuesday, let's make sure we remember to include a uncut sample. We're not going to use all of our amplomer in the digest reaction. Let's save some out and not throw it in the garbage. I think when we load our gel, we're going to want to have an uncut control in that first lane. It just seems like a good place to start on the left side of everything. So we're going to run an experiment very much like this, but for a different mouse mutation. All right, now we'll move on and talk about transcription. It's sort of in the middle of the central dogma. Double-stranded DNA can replicate. Genes get transcribed into a single-stranded RNA, and then ribosomes will take that RNA and interpret it and put together an amino acid sequence. Sometimes students are asked, why do we need this RNA middleman? Since this is pretty much the same thing. That coding strand, the one on the top, has exactly the same sequence as the transcript. It's got the same polarity, and it's got exactly the same sequence. And so that's probably one thing that, to make sure we all are in agreement about. This is a double-stranded DNA, and if we are going to transcribe one strand on that, this RNA molecule is exactly the same as this section right here. Therefore, this strand on the DNA is called the coding strand. Whether or not that's a smart name, that's what everybody calls it. Remember when we said DNA is always double-stranded? That's our assumption. But no one takes the time to write down both strands. If you write down one strand, as most everybody will, it's the coding strand that you should write down. That saves your reader a lot of trouble. The coding strand is exactly the same as the transcript. Same polarity, same sequence, except T's that were in the DNA is understood will be used in the RNA. The other strand, I think the best thing to call the other strand is the template strand. It is the strand which is used as a template by the RNA polymerase to create the transcript, but notice how it runs backwards. It is complementary to the transcript. If that's the strand of DNA we wrote down, our poor reader would have to turn it around and change every base, and no one wants to go through all that. So when we write down a DNA sequence, of course we assume it's double-stranded, but we will write down the coding strand. Oftentimes you guys are told, give your answer in terms of the coding strand. There are several other answers that are possible, but we're trying to find out if you know what the coding strand actually is. So answering the question, what is the significance of transcription? Why do we need this RNA middleman? And I think about the best way to answer this is it allows for amplification of signal. In a prokaryote, there's just one copy of every gene. But if you can transcribe that gene many, many times, and if each transcript can be translated many, many times, we have the opportunity to amplify that single copy signal that a prokaryote might have. Eukaryotes is not much better. We've got two copies, one from mom, one from dad, but we can transcribe a bunch and then translate each of those a bunch of times and have the opportunity to amplify that signal. So that's one significant, important aspect of why it's good that transcription exists. The other thing that we kind of like to have transcription for is what the whole last third of the course is really about. The idea that one gene might be on and produce a lot of product, but we might decide that another gene, we don't want to transcribe it much and we don't want to make very much product. Sometimes this even happens to the same gene, but in two different cell types. Gene A in a skin cell is getting loads of transcription, loads of translation, but gene A in a brain cell, nothing's happening at all. So that's what we call regulation, this opportunity to have a gene on, therefore lots of transcription, lots of translation, lots of function, or we can have a gene off, maybe in a different cell type or maybe in response to some other environmental change or a signaling change in a person's body, something like that. So what is the significance of transcription? It allows an opportunity to amplify the signal from a single copy of a gene up to many, many thousands of copies of a protein, perhaps. And part B, it gives us a chance to regulate the level of gene expression of most any gene. If you want the gene off, don't transcribe it. If you want the gene on, transcribe it like crazy. Now, what leads to that binary decision is what the last portion of the course is about. So we already said DNA replication, semi-conservative, bidirectional, symmetric, semi-discontinuous. We're going to use different terms when we describe these characteristics, but describing now transcription instead of replication. Maybe I can do it with some simple diagrams. So if that's our starting DNA, when we go to transcribe that, we have to locally and temporarily separate those strands. We will call that location a promoter. And for now, we can define that as the place where transcription starts. We're going to broaden our definition of a promoter as time goes by. But that's not the same term we used when we talked about where replication starts. So we've got to keep that vocabulary separate. Let's imagine that the bottom strand is my template strand. I will make a copy, an RNA copy of that strand. And a couple of seconds later, we will see that our transcription bubble has kind of moved down the street. We started transcribing here, now we're continuing to transcribe there. And then we get to the end of the gene, and we're done. We've got our double-stranded DNA, and we've got our single-stranded RNA copy, our transcript. So, it's fully conservative, isn't it? The same DNA we started with, we have at the end. The pairing arrangement's the same, nothing's been lost, nothing's been altered about it. So completely conservative. Plus, we get this new RNA molecule. Unidirectional. With respect to the promoter, this process has gone in exactly one direction. Sort of downstream. It's asymmetric. We have made a copy of this template strand and we have completely ignored the other strand, which is called the coding strand, but it's the one that we've completely ignored. It's called the coding strand because it's exactly the same sequence and polarity as the transcript. It's a continuous process. Once we start transcribing, we just go. We don't back up and start again and back up and start again. We start and we finish. That's different from replication as well. When did we finish replication? when the whole genome was done. What are we doing here? We're transcribing one gene. How often did we replicate? Once per cell cycle, the whole genome, once. How often do we transcribe? Well, if gene is what we call on, we might transcribe it many thousands of times a minute. Boom, 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 boom. Where does replication take place? In a eukaryotic cell, in the nucleus. Where does transcription take place? In the nucleus. But the product of transcription goes out to the cytoplasm. That's a change. So there's many, many differences between replication and transcription. These four terms and these four terms, these are the big ones. We've mentioned others before. DNA replication is high fidelity, RNA transcription is low fidelity, 1 mistake per 1,000 bases, which is like every transcript's got a few mistakes, that's a lot. Yeah, there's gonna be T's, where instead they'll be used for transcription. It's like the Olympics. You get a score for doing a dive, but part of the score is how hard the dive was and not just that you did it right. So sometimes we say to students, tell me 4 things that are different about replication and transcription, and you get T's instead of U's. Well, that's not a very difficult dive. Difficulty low, but you got the answer right. So we're kind of looking for these words. They have the degree of difficulty, and it gives you an opportunity to use vocabulary that you learned around here. That's what every question is. It's an opportunity to demonstrate your ability to use this vocabulary. How long does DNA last? Forever. How long do transcripts last? 2 minutes. So big differences in these two molecules. So it certainly would be a shame to confuse the very specific terminology that's been developed to describe these different processes. Then sometimes I get so hung up on the differences, I forget that they have anything in common. And then on the test they say, what do they both have in common? I'm like, nothing. I have no idea. So they both require a template. You're copying that template in an anti-parallel direction. There's stuff, there's that ribose sugar. The nucleotides are very similar. There's a lot. We polymerize always in a 3' direction. They both happen in the nucleus. There's a bunch of easy things that are the same, but I completely was stymied when I was finally asked that. So I guess we're in chapter 7 now, talking about transcription generally. They're showing what I just showed, that little bit of local temporary melting as the RNA polymerase progresses in one direction. The template and the coding strand snap right back together, leaving us exactly where we started. The single-stranded RNA gets released. Only some of it here, as it's being produced, is hydrogen bonded to the template, and then it lets go and is pushed aside by the DNA wanting to hydrogen bond with the template strand more so than does the transcript. We're only copying one strand, we're ignoring the other, so all those 4 terms can even show up in a simple generalized diagram like this. Now the next thing we want to talk about is general differences between prokaryotes and eukaryotes. We did the same thing with replication. That's the only diversity that we really have to keep track of. So, another picture from chapter 7. They are showing RNA polymerase. There's a place called the promoter where transcription will start. There's a place called the terminator where transcription will end. And so we do have a beginning and a middle and an end to this process. There is the RNA polymerase, and there is something called sigma factor. It's sigma factor that finds the actual promoter sequence. In fact, it takes sigma plus RNA polymerase in a prokaryote to find the promoter and actually stick to it. Once sigma brings RNA polymerase to that site, and it's sigma that touches the promoter sequence and identifies it, then when the double-stranded DNA opens up, transcription begins, sigma gets released. That allows RNA polymerase to take on what we call the high mobility configuration, and it zooms down the pike, creating that transcript. Until it gets to, again, a signal, a particular sequence at the end of the gene unit, and the formation of that signal tells the RNA polymerase to stop, release the transcript, fall off, go find sigma, and maybe do that whole thing again. Now, in a prokaryote, there is only one RNA polymerase, and so its name is RNA polymerase. There are several different sigma factors. It turns out a happy bacteria will make maybe the regular happy sigma, and it will lead RNA polymerase to the promoters of all the genes that you ought to be expressing when you're under good, happy conditions, well-fed, all that kind of thing. There are other sigmas that get studied. One sigma that gets studied quite easily is called a heat shock sigma. If you put your E. coli, for example, at 42 degrees instead of 37, a different sigma is expressed, and it brings RNA polymerase to a different set of genes, the set of genes that you would need to express to survive this hot situation. Viruses are famous, viruses that prey on E. coli. They will come in bearing their very own viral sigma, and their viral sigma will outcompete the cellular sigma and force the cellular RNA polymerase to transcribe not the cell's genes, but come over here and transcribe all the virus genes. So it's a nice way that a simple bacterium can have several different patterns of gene expression for a variety of conditions. Maybe starvation might be one, certainly high temperature, probably low temperature might be another one, a handful of different situations that the cell might encounter. And certainly viruses will always come in with their own sigma to try to lead the cellular RNA polymerase astray. Let's look closely at a promoter. On that, let's see, the top strand would be the coding strand. There is the beginning of the transcript. Notice how the transcript has the same sequence and polarity as the coding strand, except where there's a T, down in the transcript we have a U. We call that first base that is in the transcript +1, and then +2, +3, etc., etc. As we go backwards, this is -1, -2, -3, so it's kind of like a number line that you had back in elementary school, except there's no 0. As you go upstream, is another way we say that, around -10, most every prokaryotic promoter will have an AT-rich segment like this. And sometimes this is called the -10 box. You may have heard of the Tata box. This is not it, okay? As soon as you say Tata box, you're talking about a eukaryote. So if we are talking, as we are here, about a prokaryote, yeah, it's AT-rich, but it ain't a Tata box. This is a -10. At -35, you see another very consistent sequence that they've turned green up here. And this is what you find at -10 and at -35 in most any bacterial promoter that will be recognized by sigma. These are called consensus sequences, and I think this is the first moment where we've seen this. Consensus sequence. You wonder, gee, what is it that identifies the beginning of a unit of the DNA that ought to be transcribed? And so you look at the beginning of 100 different prokaryotic genes, and you see if they have anything in common. And all these bases seem to be not so much shared by these 100 different promoters that you might be surveying. But the ones in green are the ones that show up over and over and over again. So that's the consensus sequence. The hypothesis is this seems to be required to make this section of the gene the promoter. Then you test that hypothesis. What do? If that sequence is absolutely required, let's change some of those A's and T's into G's and C's. And if my theory is right, if that's an important characteristic of a promoter, this should no longer function as a promoter as I mutate different bases. So that tends to be the second step. So step 1, trying to figure out how a function gets carried out, look for consensus. 2, mutate that consensus. Does your function go away? The third step, I think, is one you should probably know about as well. It's called a footprint. So based on consensus And mutation now, we have the hypothesis that those sequences highlighted in green are required for promoter function. We think that means sigma protein can stick, can hydrogen bond with those particular base sequences. If that's true, then the sigma protein ought to sit right on this situation here. So if I put this sequence of DNA in a test tube, if I throw sigma protein in the test tube, my hypothesis is when that sequence is there, sigma will hydrogen bond with it. To see if that's true, I throw in an exonuclease. So endonuclease cuts DNA in the middle at a particular site, if it's a restriction endonuclease that we're familiar with. An exonuclease will just nibble away at the ends. So I throw the exonuclease in, and it will nibble away at all the double-stranded DNA that's in there, except the double-stranded DNA to which sigma protein is bound. Sigma protein makes a footprint. It covers up the DNA so it can't be digested. So that's usually the third thing that happens. So considering how does DNA sequence carry out a function? Well, first of all, get evidence that there is a consensus sequence that is circumstantially associated with the function. Change that sequence and show that it is required for that function. And then identify the protein that footprints that sequence, and you're really demonstrating that that protein is seeing, sticking, hydrogen bonding with that sequence and only that sequence. And this 1, 2, 3 approach to unraveling molecular biology has been used forever and ever and continues to be used. So that's probably an approach that you want to be thinking about. So sigma, carrying RNA polymerase on its back, comes here, opens up that complex, will open up the double-stranded DNA. It'll start to transcribe and lay down bases here. Sigma will be ejected. RNA polymerase will take on the high mobility configuration and transcribe the whole length of the gene. So we're skipping the middle, and here we are at the end. So we're transcribing. Again, notice we've got a perfect match for the coding sequence. And we're transcribing, and we're transcribing, and we're transcribing, and then we stop. And what they noticed, again, well, is there something about the termination sequence that's the same in every gene that we study? So find 100 genes, look at their tail end, see what all of these transcripts end with. And they come up with this and this. And you look at that for a few seconds and you realize it's a palindrome? G, C, C, G, C, C. Okay, so what can a palindrome do? So as that coding strand is transcribed into this, what RNAs want to do is form secondary structures. We think of them as just spaghetti strings, but they take on secondary structures. And so when this, as soon as it's created, it will form a little double helix with itself. And quite stable. Look, GC, 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 GC. A lot of GCs are going to make that little lollipop shape be quite stable. Well, the minute that forms, remember RNA polymerase is only sitting right here in a position where it can contact this newly created shape. I think of that as a little finger, and it's tickling the back end, the butt end of the RNA polymerase, and it makes it giggle and let go and stop the whole process. And I think the giggling and letting go and the butt end is all funny to me, but these three-dimensional interactions between proteins and nucleic acids, shapes and shapes, hydrogen bonding, this is the kind of theme that we'll see over and over again. And so the terminator sequence seems to be a palindrome, which at the RNA level can form this little so-called terminator hairpin. And then that seems to be followed by a string of U's. U's will only form 2 hydrogen bonds with the template strand, and therefore it seems like it's real easy to make it let go, so that seems logical. It's just like the AT rich -10. That must be the place where the DNA first separates, because there's the smallest number of hydrogen bonds there. It must be kind of loose and able to unzip it at that point. Okay, so for a prokaryote, we've got a promoter, we've got a terminator, we've got a single RNA polymerase, we've got choices of sigmas that will give us slightly different genetic programs. And that's kind of the whole story. Now, eukaryotes are a living nightmare. There are 3 RNA polymerases. And they go by the name Roman numeral 1, Roman numeral 2, Roman numeral 3. Now, why this is important is because when we talked about replication, we said prokaryotic DNA polymerases go by Roman numeral 1 and Roman numeral 3. So they love to design questions to see if you kind of remember Roman numerals, but you forgot the context. Well, Paul 1 and Paul 3, that was prokaryotic DNA replication. Now we've got Paul 1, Paul 2, Paul 3. That's eukaryotic transcription. If there's confusion between those two situations, they do not want you in medical school with a scalpel in your hand. That's the sort of reason they're trying to see if you get easily confused. Did I say spleen or did I say pancreas? What's the difference? So, Paul 1 in a eukaryote is responsible for transcribing genes that encode the ribosomal RNAs, which you already know are part of ribosomes. They come in sizes 28S, 5.8S, 18S, and that's what those genes encode. Those are the finished gene products. They are just part of the ribosome. Ribosomes are awfully important, and so there's a whole class of genes and a class of promoters that is only read by RNA polymerase 1. End of story. RNA polymerase 3 encodes, transcribes genes that code for the transfer RNAs, the tRNAs, which are tiny, and also one of the ribosomal RNAs, which is 5S, the smallest of the ribosomal RNAs. Okay, and that's kind of the end of that story, except I don't know if it's news to you guys. There's more than mRNA, tRNA, and rRNA these days. These days there are snRNA and miRNAs as well. And so that's another thing, another class of genes that RNA polymerase 3 is devoted to transcribing. But the star of the show is RNA polymerase 2. RNA polymerase 2 is the one that transcribes the genes that we think of as genes, the protein coding genes, where the final gene product is not the mRNA, but rather the protein product that is transcribed following the mRNA instructions. In fact, when we count genes, when you read that humans have 20,000 genes, we are only talking about the protein coding genes. We are completely ignoring the hundreds and thousands of genes that encode